Thanks very much, everyone, for coming along, and welcome to the next Data Science Under the Hood seminar. So for those that don't know me, my name is Chris Vandy, and I'm one of the program directors in the Centre for Data Science. So it's a great pleasure today to introduce Professor Matt Simpson as our speaker. So Matt has been awarded many ARC grants, including some prestigious fellowships, such as the Future Fellowship. And he has also won some prestigious awards in Australia, including the J.H. Michel Medal and also the E.O. Tuck Medal. So Matt is a world-leading expert in mathematical biology. He has developed new deterministic and stochastic models in developmental biology, and also developed methods for connecting these models to data. And more recently, Matt has been working at the interface of mathematical modeling, uncertainty quantification, parameter estimation, and identifiability analysis. So Matt's presentation will go for around 30 minutes or so, after which there will be plenty of time for questions. So if you have any questions as the talk proceeds, then please don't hesitate to type them into the Q&A window, and then we can look to address those questions at the end. So now I'm going to pass it over to Matt, who will talk about likelihood-based methods for model selection applied to coral reef regrowth. So thanks very much, Matt.
Thanks, Chris. Thanks for the kind introduction. Thanks to the Centre for Data Science for inviting me to talk. What I'm going to talk about today is combinations of mathematical modelling and problem solving using ideas from data science, and that's my real passion in my research activities here at QUT. And the work that I'm going to talk about really started off with some collaborations with my former PhD student, David Warren, who was looking at using mathematical models to study the regrowth of coral reefs after some kind of damage. So I guess here, for most of us in Southeast Queensland or in Australia, we know about the Great Barrier Reef and the threats that, populations of corals are facing. And what we're trying to do is to use some mathematical models to try and understand these population growth processes. Okay, so the data that I'm going to work on today is just one, in some sense it's a very small data set, but in another sense it's a very large data set. The data that I'm going to talk about is going to describe the regrowth of the coverage of hard coral, shown here as a plot on the right-hand side, over time after some disturbance. So this is a publicly available data set, and this coral population here is close to Lady Musgrave Island, which is off the town of 1770. It's in the southern part of the Great Barrier Reef. And one of the reasons that we're interested in this data set is to think about what happens after the coral cover has been disturbed by some event. So it could be a cyclone, or maybe some kind of bleaching event, or some runoff event. And what we've got here is some estimates here of the percentage of hard coral cover over time. And you can see this is measured in days after some disturbance. So in some ways, the data set is small, and that we're looking at one coral reef recovery, but in other sense, it's quite long. So we're looking over 4,000 days. So what we can see here over a period of 10 years is how the coral recovers after the disturbance. And we might be interested in, for example, after the bleaching that's taken place in last summer, we might be interested in which coral reefs will recover. We might be interested in how long they'll take to recover. And the question of how long they'll take to recover is really connected intimately with ideas about the resilience of the coral reef. So if the time taken to recover is shorter than the time between disturbance events, then we might expect the coral reefs to survive long term. However, if they're going to take longer to recover than the time between events, then we'd have grave concerns for those reefs. And one way to do this is to use a mathematical model. So really what I'm going to talk about is mathematical models of population dynamics, and mathematical modeling and population dynamics, biological population dynamics, is probably one of the oldest areas of applied mathematical modeling. Going back to 1798 with the proposition of the Malthusian growth model, which is the well-known exponential growth model here. Here I'm talking about some concentration or some population size C. It's going to grow exponentially at some rate R. And there's many other extensions of mathematical models, probably of population dynamics. Probably the most famous, known and well studied is the logistic growth model here. First described by Verhulst in 1830s. And what we've got here is the addition of some factor which is going to slow the population rate growth down as the density or the population size gets towards some carrying capacity. And the logistic growth model has been used and rediscovered in many different applications, and it's been attributed to many people after Verhulst, including people like Pearl, Reed, Lotka, and McKendrick. If we think about these two population growth models here, the exponential growth on the left-hand side looks like the early part of a growth process, like the early time in the coral reef recovery. We've got the rate of change of increase being proportional to the population size. And the main difference with the logistic growth population here is that over time, we get a sigmoid response here where we've got approximately exponential growth at early times. And then as the population size reaches some carrying capacity, then the population growth slows down. So we've got some asymptote. And this is a classical observation in population biology. And one of the challenges that we face when thinking about how to model populations is that there is a massive catalogue of this kind of S-shaped sigmoid growth curves. And one of the challenges we face is what type of model do we use to model particular situations? So I first started showing you some data from Lady Musgrave Island, which had that classic S shape here. And that's a large scale, kind of hundreds of kilometers in scale and decade over time scale growth process. We also see similar S-shaped growth processes at very different scales. Here are some experiments generated by mathematicians in my lab working on cancer biology. This is an image here, 3 images shown over time of a population of cancer cells in a little tumour growing over time. And indeed, when we look at the size of these spheroids, these radial spheroids, and we plot them over time, one thing we see this S-shaped growth and this saturation to a carrying capacity. In this case here, what we see is different tumours starting at different sizes grow up and asymptote towards the same carrying capacity, and that's consistent with the logistic growth model. It's also consistent with many other mathematical models. So not only do we see this kind of S-shaped growth taking place in tumour spheroids or populations of tumours grown in vitro in glass, we also see the same kind of ideas happening in vitro in tumours. So this is a paper here published about 20 years ago in Nature, where an idea was to look at the growth in time of the size of tumours in different types of animals. And these authors here went to a lot of effort to make the point that perhaps you could come up with a universal population growth curve by looking at the size of tumours in pigs, rabbits, rats, guinea pigs, and shrimps, and so on. So this is a very active area of research. While this paper was published 20 years ago, this idea of looking for a universal law or universal growth law in biology continues. But modelling in biology is not like modelling in engineering or physics, where we've got access to well-known laws like law of thermodynamics or the laws of motion. In biology, the laws of biology are much more elusive. So it's a very active area of research, but in general, we're not sure about which model to use in which situation. So what I'm going to talk about today is some very simple looking mathematical models. These are the type of mathematical models you might have seen in high school mathematics, or you'd certainly see in my first year engineering calculus course. So we're going to look at an ordinary differential equation. dCdt is R times C times some crowding function. So if we set the crowding function to be 1, then we get exponential growth, and we've got a very, very simple model. But when we look at different types of models in the literature to capture this exponential growth curve, they really all differ in the choice of this crowding function f. And what I'm going to show today is just compare a few different models. So if we look at the logistic growth model here, what we've got in the crowding function here is just a linearly decreasing function of density. It's going to linearly decrease between 1 and 0, and it's going to reach 0 when the density hits the carrying capacity. And that's what gives rise to this differential equation here. And this differential equation's got a nice exact solution. You can probably, students in my first year calculus class would be computing this exact solution. But when it comes time to thinking about applying this mathematical model to data, if we think about the parameters that we've got to estimate, we've got a growth rate, R1, a carrying capacity density, I'm going to call K, and you probably also want to estimate the initial density, C0. The crowding function is a log function here. So when we put the logarithmic function into the ordinary differential equation, we get this differential equation here. And again, we can solve this differential equation exactly using first year calculus. And when we come to apply this differential equation to data, we'd be looking at estimating the growth rate, R2, carrying capacity, K, and also an initial density, C0. And there's a massive catalogue of these types of models. What I'm going to do is present one more model called the Richards model. And the Richards model is an extension of the logistic growth model here, whereby we've got an extra parameter, this exponent sitting here in the crowding function, and it's an extra parameter that gives us a nonlinear decreasing crowding function. The differential equation here also has an exact solution, and with this extra parameter, we've got one more parameter to estimate when it comes time to thinking about applying this model to real data. When we apply these models to real data, it's worth noting, and I won't dwell on this, but I'm happy to talk about it later, that all three of these models are structurally identifiable. So what that means is there's only one piece of information you might collect to characterize these growth processes. That's observations of the density C, or the coverage C. And so by being structurally identifiable, this means that if we've got perfect observations of C, then we can always distinguish between these three models here. But of course, if we think back to the data at Lady Musgrave Island, we've got discrete observations and then noisy observations. So we're interested in what we call practical identifiability, where we're working with finite amounts of imperfect data. So I'm going to focus on these three models in the talk today. And I just thought I'd start off maybe taking a bit of a mathematician's perspective by thinking about what the difference between the models are. So I tried to summarize these differences up the top here algebraically, and then I'm showing some plots underneath. So the models here all differ in this crowding function. So the logistic model has a linearly decreasing crowding function. So here I'm showing the crowding function decreasing as a function of the density. And of course, that's going to go to 0 when the density hits on the carrying capacity. The Gompertz model is a decreasing function given by this logarithm here. So it's going to decrease and also pass through 0 here when the density hits the carrying capacity. And the Richards growth model here has got a family of growth curves that depend upon this exponent beta here. So here I've shown about half a dozen crowding functions here for the Richards model, and these are shown in order of increasing values of beta. In each case, the differential equation can be solved. It can be solved exactly, and I can plot the solution, giving rise to these sigmoid growth curves down here. I'm plotting the solution in terms of dimensionless time, and I'm plotting the density in terms of dimensionless density. And so here's the solution of the logistic curve shown in a dimensionless format, the solution of the Gompertz here shown dimensionless format, and several solutions of the Richards curve here shown in terms of dimensionless quantities for different choices of this exponent beta. And we can see down here that they all capture similar behavior. They all capture this sigmoid curve. The question we want to answer is, which model is best for our data? But before I do that, I'm going to stick with a mathematical perspective for the moment and just think about the relationships between these models. So these models are qualitatively related by all capturing this sigmoid behavior, but they're also quantitatively related. If I just scroll back up for a moment here, if you look at the crowding function in the Richards model, I think it's obvious that if I set this exponent to be 1, then the Richards model collapses exactly to the logistic model. So here what I've done here is to sketch the sigmoid curves. I've sketched a range of sigmoid curves, and I've shown results for different values of this exponent beta. And in each subplot here, I've got a red curve for the Richards model, I've got a green curve for the Gompertz model, and a blue curve for the logistic model. And over here, when beta is equal to 1, then the Richards model and the logistic model exactly coincide. And I think that's obvious from looking at the mathematical models. What's less obvious is when we look at smaller values of beta, so beta is equal to half, beta equals 0.3, beta is equal to 0.1, you can show mathematically that the Richards model tends towards the Gompertz model. And you can see that here that we're getting closer and closer. Okay, this is a good example. If I first came to this problem of thinking about which model will I choose, I think, in my opinion, this is a good example of somewhere in mathematical modeling where theory has really outpaced practical problem solving. So here I'm just showing a screenshot of one of my favorite mathematical modeling papers. This is a nice paper written 20 years ago now. It's actually a summary of a student's master's thesis in which the authors really lay out a whole host of logistic growth models, lots of generalizations of growth models, something like a dozen different variations of a logistic growth model. These models have all got names, they're named after people. And in this paper, what these authors do is summarize all of these models together. They summarize a range of exact solutions. They summarize a range of mathematical properties, and it's a little nice excursion into special functions, and this is great. This paper has been cited a lot, but one of the things you'll notice from a practical point of view, if I want to ask the question, how long does it take for coral at Lady Musgrave Island to grow back? This very successful paper does not mention how to connect any of these models with data, and I think that's a very important question for us to think about. Okay, so what I'm going to do just in the next few pages is to explain how we connect the mathematical models with data. And this is a very standard likelihood-based parameter estimation exercise. So first of all, what I'm going to do is thinking about the predictions of my model at different values of time for different choices of the parameters. And I'm going to represent that with the variable y subscript I. So that's going to be the predictions of the mathematical models. I'm then going to assume that I've got my observations collected over different values of time. I'm going to give that variable y for the density, and I'm going to give a subscript O to think about observations. So my observations, given some parameters, I'm going to assume that they are normally distributed around the solution of the mathematical model. And since I've got a very simple noise model here, I'm going to assume that I've got a constant variance. All of the work that I'm going to talk about today involves having a Gaussian or a normal noise model, but everything that I'm showing you can be repeated with different noise models. So if you prefer a binomial or a negative binomial model, you can repeat all of the analysis that I'm about to show you today. The point I'd like to make is that once we've got this noise model here, we've got an extra parameter that I'm going to estimate the variance or the standard deviation in the normal model. In my time series of data, what I'm going to do then is to construct a likelihood function. So here's my likelihood here. I'm going to assume that the data is independent. So I'm going to sum up over time series of observations. I'm going to compute the normal PDF, and I'm going to take the logarithm. So I've got a log likelihood function here. And so phi here is the density of the normal noise model with the mean of my observations, and I've got a constant variance here. And once I've got this likelihood model, this likelihood function for each of my different models, I can then find the parameters of best fit simply by using maximum likelihood. So I'm going to use numerical optimization. All the work I'm going to show you is performed in MATLAB, so I'm really just using something like fmin_con to estimate the parameters. And this is a very quick and efficient calculation. When I take the data that I looked at before for the regrowth at Lady Musgrave Island, I can estimate the parameters in each of my models, the logistic, the Gompertz, and the Richards model. And I'm not going to lay them out in a table here. I can give you a reference at the end that kind of gives an exhaustive description of these models. But what you can do is you can compare the best fit solution of the mathematical models with the data, And you can at least compare by eye that, yes, this looks reasonably good. You can also do some extra model checks if you like. You can analyze the normalized residuals and check that they've got a mean of 0 and that they're approximately normally distributed. And we find that in each case, each of these models seems to do a reasonable job based on these simple criteria. However, if we look at a practical implication of this parameter estimation, what I'm interested in is looking at an estimate of the timescale for the coral regrowth. So in each of the estimation procedures up here, I've got an estimate of the growth rate, R1 for the logistic model, R2 for the Gompertz model, R3 for the Richards model. If I take 1 over those growth rates, that gives me an estimate of the timescale of regrowth. And if I take my best fit parameter estimates and I compute 1 over the regrowth, then that tells me I get a finite estimate of the duration of time, or a characteristic time of regrowth. So that tells me with the logistic model is going to predict 400 days. The Gompertz growth is going to predict a characteristic timescale of 630 days, and the Richards model is going to give us a characteristic timescale of regrowth of 180 days. And clearly, those numbers are all quite different. They're not just out by a factor of 10% or something. They are quite different predictions, and that really motivates the question of which prediction should I believe. In order to probe further into that, I'm really going to delve now a little bit more deeply into distinguishing between these models using an identifiability analysis. So what I'm going to do is to work with a normalized likelihood. What I'm going to do is take the likelihood and subtract off the maximum value. So the MLE for each model has a normalized value of 0. Then what I'm going to do is to think about assessing the practical identifiability of the different parameters in the model by taking the parameters and splitting it up into an interest parameter psi and some nuisance parameters lambda. And I'm doing this so I can look at the profile likelihood. And I'm going to construct the profile likelihood again using numerical optimization. I'm going to look at the profile likelihood of my interest parameter or my interest parameters simply by optimizing out or maximizing out the nuisance parameters. And that's going to turn out to be very useful for me to really look into the likelihood function. Remember, the likelihood function, or the normalized likelihood function, is perhaps some relatively complicated function of 4 or 5 parameters, maybe more for more complicated functions. So it's a difficult function to visualize. And the profile likelihood gives us a way to systematically look at this, to analyze this complicated likelihood function. So to make this a bit more concrete, if I think about taking the logistic growth model here, my parameters are the growth rate, the carrying capacity, the initial concentration or the initial coverage, and here I've got the standard deviation, or I could have the variance in the noise model. If I'd like to construct a profile likelihood of the growth rate only, I would select the R1 as my interest parameter, and my nuisance parameters will be all of the remaining parameters. And then what I can do is to construct a profile likelihood by optimizing out all of the other parameters in the problem. And if I construct a profile likelihood across a uniform mesh in R1, what that allows me to do is to look at the extent to which the likelihood is well peaked around this parameter. And of course, I can do that for each of the parameters in turn. So in the logistic growth model, I've got a growth rate. These are the plots of the profile likelihood for the growth rate, the carrying capacity, and the initial density. I also construct a profile likelihood for the variance in the noise model, but that hasn't, I'm not going to show that here today. I'm just going to focus on what I would consider to be the most important parameters.
Yeah, I think we just lost you briefly there, Matt.
All right.
Yeah,
so
it's up now. It's up now.
Okay, thank you. So what I was showing here were the profile likelihoods for the parameters in the logistic model. And I'm really focusing on the growth rate, the carrying capacity, and the initial density. And I'm not showing the profile likelihood for the variance in the noise model. I'm just focusing on these ones. But of course, you can compute that as well. What I'm showing in each of these profile likelihoods is the MLE, shown in the vertical red here. I'm showing a threshold that's related to asymptotic estimates of confidence intervals. And when we look at the profile likelihoods here for each of these three parameters, we can see that they're well formed. There's a single peak and the profiles form relatively narrow. They're relatively narrow. And so we've got good confidence in the estimates of these parameters. I can do the same for the remaining Gompertz and Richards model. So here I've shown the profiles for the logistic. These are the same as I've shown before for the growth rate, carrying capacity, and the initial density here. Down the middle, I've got the analogous results for the Gompertz model, the growth rate, the carrying capacity, and the initial density. Curiously, in this case, the initial density of the profile is quite wide. It's a bit hard to tell on this scale here, but the initial density profile goes over a couple of orders of magnitude in width. And in the Richards equation, the more complicated model, then I've got profiles for the growth rate, the carrying capacity, the initial density, and the exponent. And of course, what we can see is that we don't have well-formed single-peaked profile for this case here. So in summary, what I'd say is that each of the individual parameters in the logistic model are identifiable. In the Gompertz model, we've started to encounter practical non-identifiability in the sense that we can't quite pin down the initial coverage. And certainly in the Richards model, which is only slightly more complicated than the other two, we're having difficulties with identifiability of the growth rate, the initial coverage, and this exponent here. If I was undertaking a fairly standard model selection using likelihood, what I'd probably be doing is computing some kind of information criteria. Here I've shown what you might find as a standard formula for the maybe an Akaike information criteria, where we'd be looking at ranking these three models in terms of a single scalar quantity, and we'd be ranking them by trading off two pieces of information. First of all, this term here is going to tell us what the likelihood, the maximum likelihood is. So we're going to prefer models that fit the data well. But what we'd be doing is we'd be trading off against model complexity. So K here is going to be the size of the parameter. So we're going to rank models. We're going to prefer models that give the best fit to the data, but we're going to prefer simpler models. And if I compute the AIC for these three models, I would rank them logistic, Gompertz, and Richards, just as I've shown on the previous slide with the profile likelihood analysis, except with the simple scalar ranking, we really don't get any insight into why one model might be better than the other, or we don't get any insight into what happens beyond the other edge of identifiability. And what I'll do is just conclude my talk in the next minute or two by showing what else we can do with a profile likelihood analysis to really provide extra information. So I'm going to go back and have a little bit more of a think about the Richards model. So the Richards model is the most complicated model that we considered. The crowding function here has got this pesky exponent that we need to estimate. The full parameter involves a growth rate, a carrying capacity, this exponent, an initial density, and the variance or the standard deviation in the noise model. And if I just scroll backwards a couple of slides to the profiles here, you can see that the carrying capacity was really the only parameter that we had any great confidence in when working with the Richards model. So we had no great confidence in our estimate of the growth rate or the exponent. So what I can also do to provide extra information into the Richards model is to think about constructing the profile likelihood with different interest parameters. Here I'm going to consider the growth rate and the exponent as interest parameters, and I'm going to optimize out the remaining nuisance parameters. And so I'm going to be working with a bivariate, or I'm going to be looking at a bivariate profile likelihood here. And of course, what I can do is evaluate this bivariate profile likelihood across a mesh in R3 beta space, so simple square mesh. I can optimize out the other parameters, and then I can get an assessment, or at least a visual assessment, about any trade-off or relationship between these two parameters. So if I do that, and I look across a square mesh and a range of growth rates and a range of exponents, what I can see here in red is the maximum likelihood estimate. If I also keep track of some threshold values, so the threshold values are shown here in dark gray, what I can do is I can really capture a credible region. And of course, this region has got this classic banana shape, suggesting that there's many combinations of beta and R3 that give good matches to the data. When we see patterns like this, it might tell us that actually the product of beta and R3 is something that could be of interest. Now, this is something we can also tackle with a profile likelihood analysis. If I take my interest parameter, or if I rescale the mathematical model, so I think my interest parameter is the product of beta and R3, then what I can do is construct a univariate profile likelihood of this product. And when I do that, what I can see is the data that we've got is sufficient to identify the product of beta and R3. Draw my presentation to an end. So I'll just have a few points of conclusion and then maybe take some questions. Hopefully my internet stays stable long enough to do that. Sorry, Chris. So in conclusion, what I've done today is I've really considered some mathematical and statistical modelling tools that might be helpful for us to understand population growth in the context of coral reef regrowth after a disturbance. And this is very topical for us in terms of Great Barrier Reef management and strategies for management. Standard approaches, if you look in the literature to model coral reef regrowth, involve using a Gompertz model without considering whether the parameters in that model are identifiable or not. And I think what I've done is show you here today that this can be dangerous because simply applying maximum likelihood estimation without considering identifiability could lead one to make predictions with overdue confidence. The profile likelihood tools that I've shown here today, they really show that the Gompertz model and the Richards growth model, which is the very next simplest model you might use, are not identifiable when it comes to working with these types of data. And I've also just made a point that standard model selection based on information criteria they're very attractive, they're very simple, but they don't always provide insight into what happens when things go wrong. In contrast, I would claim that profile likelihood analysis is fast. Numerical optimization is often very fast, especially compared to sampling. It's versatile in that it allows us to consider, identify and to consider combinations of parameters. And it's insightful. It's particularly insightful for the very practical cases we're interested in, where we're dealing with a problem that's non-identifiable or borderline identifiable. Okay, so the work that I've done in this project really involves a great collaboration with terrific collaborators, Oliver McLaren at the University of Auckland, Ruth Baker at the University of Oxford, Alex Browning, who's a postdoc here at QUT about to depart and commence a fellowship at the University of Oxford, and David Warren at QUT. I've tried to keep the talk to a technical minimum today. I hope I've succeeded in that. If you'd like some more information, the work that I presented today has just been published in the last maybe a month or so ago in the Journal of Theoretical Biology, really focusing on sigmoid population growth, trying to cover all of these ideas about coral reefs and cancer populations, but really with a case study on the coral reef. All of these models here I've talked about today are ordinary differential equations. If I had my mathematician's hat on and I'm more interested in partial differential equations, then everything I've talked about here today also carries across to partial differential equation based models, in which if you find your way through the Twitter links into my webpage, you can find some other references. So that was all I was going to talk about today. Hope I didn't use up too much of your time. Happy to take questions. Thank you very much.
Thanks very much, Matt. That was a really interesting presentation. So we already have one question that's come up in the chat. So I guess so, given that you've been working with deterministic mathematical models in your talk today, I think the question is around, would you get sort of any benefits from considering a stochastic version of the model, and if that might help you to identify the growth pattern compared to, say, using deterministic models?
That's definitely the case. It's probably hard to give an answer to that that's the true of all situations, but certainly if people who are interested in using stochastic models might find it difficult to connect with the deterministic work that I've given here. One answer I'd give to that is that almost many stochastic models that we work with, you could take a continuum limit and then think about considering the identifiability of the continuum limit models. You can also look at stochastic identifiability, and people indeed do that. It's a slightly different approach, but could yield more information. So yes, there is more to life than deterministic models. What I would say is that looking at profile likelihood analogues of a stochastic model is probably even more advantageous than working with a deterministic model, simply because of the computational speed up. Working with some kind of numerical optimization is often very fast, and so optimizing some kind of likelihood or some kind of surrogate likelihood could be very advantageous, perhaps instead of a different approach like taking a sampling approach like an ABC type approach, which could be very slow to diagnose identifiability.
Excellent, thanks, Matt. So we've had another question come through in the chat window. So have you considered non-frequentist approaches to looking at practical identifiability? So maybe a Bayesian approach.
Yeah, look, I have, and in doing that, I became quite interested in frequentist approaches. So that the reference I've got here on the bottom of the slide, I think you can still see the slideshow, practical parameter identifiability for spatiotemporal models. In that study, what we did was to look at a set of partial differential equations, which nonlinear partial differential equations, and those differential equations required a numerical solution. So everything I showed you here today has got an exact solution. So once we start working with numerical solutions, then computation time becomes, I wouldn't say a concern, but it can become a concern. So what we found was, looking at an MCMC type approach to identifiability, was that for the problems we considered in that study, where we looked at simple models that were identifiable, we were able to look at MCMC chains and look at averaging out across to look at marginal densities. We'll be able to do all the usual things that you might do in an MCMC study, and show that we could find the problem was identifiable. We also found that with that likelihood function, we could just optimize it numerically using fmin_con and get the same answer. Where the profile likelihood approach really becomes advantageous is when you are on the edges of identifiability or non-identifiability. So in that case, if we dealt with a more complicated model, then you could set up your MCMC algorithm and let it run and wait for a long time, and then be very disappointed that the chains didn't mix or they didn't converge. And that's fine, because you know that you don't have a good answer, but it doesn't really tell you why, and it can take a long time. When we use the profile likelihood analog, we found that we got very wide or very flat profile likelihoods, or we got one-sided profile likelihoods. And so we were able to identify non-identifiability much quicker. So by much quicker, I'd say an order of magnitude. Instead of hours of waiting for an MCMC chain to converge, we would be able to do some numerical optimization in maybe a minute and get the same answer. And what I really like about, what I'm really enthusiastic about working with profile likelihood, is that we could then reparameterize the model. So we might be able to look at, just like I did today with the product of two parameters, we were able to look at, were two parameters in the complicated model equal to each other? So for example, we could profile the difference between two parameters and show that it was close to 0, or we could profile the ratio of two parameters and show that the ratio was close to 1. Now that's really insightful, because it not only tells you that the problem is not identifiable, it tells you how it can be not identifiable. And I'm sure there's plenty of experts working in non-frequentist methods that could tell me there's an analog of doing that in the non-frequentist world. But from my point of view, I thought the frequentist approach was very attractive for looking at what happens when you step across the borderline into a non-identifiable problem.
Awesome, thanks, Matt.
Thanks, Chris.
So I do, so if there's any more questions, please don't hesitate to type them in. Actually, one just popped in.
Yep.
Okay, so this is another interesting question. So if you're using the models for forecasting, what model from those you analyze would you use and why?
That's a good question. Thanks, Matthew. Look, based on what I've shown you today, I would say you would be using the model that is, well, I'd say what you do is avoid models that look like they're encountering practical identifiability issues, because you might have a maximum likelihood, but of course, if the likelihood is flat, then you might be making a prediction without great confidence. That's a simple way to answer that question. Other work that I'm focusing on more recently is to then look at giving prediction intervals using profile likelihoods, and really using the profile likelihood as a tool to study which parameters affect different parts of the prediction. And I feel like, so in what I've shown you today, I think the simplest answer is steer away from a non-identifiable model. But if I was to give you a different talk down the track where I'd finalise some current results, I would say to you there are other ways to use the profile likelihood analysis to look at prediction intervals and parameter by parameter influences on those prediction intervals. And I think that's a more satisfying answer, but that's still work for me that at least is ongoing. So I think there's work in this space.
Thanks, Matt. Yeah, it definitely makes sense. So I do have a question if I can fit one in. So the quantity of interest really for your application was the time until regrowth or something.
That's right.
And so is this just like a function of the model parameters?
Well, it is. That's right. It's a function of the model parameters. So the simplest, the only parameter in these models that has, if I just go back and look at the only parameters in these models that has a dimension of time is the growth rate. So if you look in a biology experiment, for example, people will look at estimating the doubling time. That would be related to 1 over or be proportional to 1 over the growth rate. The other parameters don't explicitly involve time, but of course they're coupled together in a nonlinear model, so you can't estimate one without the other.
Right.
But so really the estimate the parameter you'd be keenly interested in is the low density growth rate.
Right. So I was sort of just wondering if it was possible to construct a profile likelihood for that particular quantity if it's just some function of the model parameters, and then you might prefer the model that gives you the best identifiability of that particular quantity.
That's a good point, and that's also another area that people in the profile likelihood literature work on in terms of adding additional constraints in terms of some kind of prediction. So yes, that's there is some work, although it's, as far as I can tell, it's not very well developed work in the profile likelihood literature that really focuses on some quantity of interest and then looking at intervals of that quantity of interest. So that is also something that that could be an extension of this work. Yes.
Excellent. So I think I had one last question. So sometimes people do, when they're doing predictions, they do sort of model averaging. So they take a few different models and get a score for each of them and weight them in some way.
Right.
So can this sort of framework be extended to accommodate that, or is that something that you wouldn't sort of recommend doing?
It would be hard for me to recommend that, Chris, because I haven't thought about doing it, but it's certainly something that I think could be investigated, and maybe that's something that, you know, if people are interested in that, I'd be happy to chat with them about those kinds of ideas. So I've not thought about how to combine models like this. I mean, my first approach has been to do the simplest thing first. But yeah, I think that'd be a really useful strategy in terms of thinking about how to combine, you know, nice features of multiple models, or maybe to avoid less nice features of multiple models. I'd be very keen to explore how that might work.
Yeah, okay. All right, thanks very much, Matt. That was a really interesting presentation. Yeah, it was sort of really nice to see an example where you've got several models that sort of fit the data equally well, and there's a massive question of which model do you choose. And I really enjoyed the way that you presented a nice solution to that, is to focus on the model that you can identify properly. And so that was really nice. So thanks very much for that.
Thanks, Chris.
Yeah, so I think we can bring the session to a close now. So let's thank Matt again for a really great presentation. And thanks very much, everyone, for joining today. It's a really great turnout, lots of really interesting questions. But yes, I hope to see you again next time for the next Data Science Under the Hood seminar. But it's bye for now.